So we're going to take a look at the production of proteins. Look, it's almost 11 o'clock. Wow, the last 2 hours really flew by. I hope the teacher didn't say anything important. I'm sure we all have days like that. Okay, we're going to start with some structural stuff, but just some reminders. Stretches of DNA that are codes for particular protein types are genes. Variations on those sequences are alleles. They're generally stuck together on much larger stretches of DNA, which are chromosomes. In eukaryotes, especially in people for sure, a tremendous amount of the DNA in the chromosomes are not codes or are not used codes. What they originally called junk DNA, because we're people and it's like, well, we don't know what it's doing, must not be doing anything. We're finding out that there's a whole bunch of stuff in junk DNA. We've talked about that earlier in the semester, that some of it's old code, some of it's foreign code, some of it's codes for RNA that we didn't know there was such a thing for when this was first kind of settled upon. A lot of it, though, has to do with this stuff. This is the structure the DNA takes in typical cells, where it is wrapped around these spooling proteins called histones, which they have represented here as little balls. That's close enough. And then, so it coils on them, and then it packs in the coils, and then it makes coils of the coils, and then those all clump together. And in the nucleus, in interphase, it would be in this shape. In late interphase, and actually in prophase, it would start to pack together into what would be a visible chromosome under a light microscope. And remember, this is copy connected to a copy, sister chromatids. And this is this says that this is a transmission electron micrograph of a chromosome. You can see this stuff if you get a good enough electron microscope. You can actually see the histones and the stringing together these clumps. Well, another representation. There's only a few different types of histones. They tend to be remarkably similar across a very wide swath of living things. You can see here they're showing kind of different ways of how it packs together. We're not going to get that far into that. Just a reminder, a eukaryotic chromosome is two-ended. A prokaryotic chromosome is a loop, but it still has histones. So histones are something that you find in basically all cells wrapping up the DNA. In prokaryotes, they don't have a nucleus, but they do have an area where the chemistry to process DNA is a little bit more localized and specialized, called a nucleoid. It's just not sealed up within a membrane. And when you need to get at a code, you need to unwrap that part of the DNA in order to get the code off of it. In prokaryotes, because they're not in a chamber, typically in a eukaryote, the need comes in from outside the nucleus. So you find the gene, you get the code, you bring it out, you make it out at a ribosome. And so there's a number of kind of nonlinear steps there. In prokaryotic chromosomes, it's very common for you need to make something and it's going to take 5 steps and it's going to require enzymes for every step. The genes for those enzymes are going to be right next to one another on the chromosome, so that you can make the enzyme and do the processing and make the next enzyme and do the next processing. So the chemistry and the codes are much more closely associated. And it makes sense to put pathway enzyme codes in a sequence on the chromosome. So you open the same stretch of chromosome, make your little pathway, get it done. If I want to do the same thing in a eukaryotic cell, I got to get the first enzyme out. I got to get the second enzyme out. Got to get the third enzyme out. It doesn't really matter if they're in the same location on the same chromosome. For all I know, it may be easier to go to different chromosomes in the nucleus and get the codes separately. But you don't tend to find genes linked in sequences that way. Developmentally you do, but that goes back a long way. Also, don't forget prokaryotes also will take sections of chromosomes and put them on a plasmid. And sometimes a plasmid will have a sequence of genes involved in a pathway on it as well. We've talked about telomeres and telomerase and centromeres and chromatids and all that kind of stuff. And this is all building up to the basic process here. So what do you do with the code? The code is in there on your DNA. It's on just one side. The other side is the duplicate that allows you to keep making copies of everything and distributing them properly. This can't get out of the nucleus. You need to put it onto a different nucleic acid that can leave the nucleus, and that would be RNA. It would be messenger RNA. I'll go through this and write it down in a second. Messenger RNA can leave the nucleus, and it's going to go out to a ribosome where proteins get made. It's going to feed through the ribosome, and as it feeds through the ribosome, amino acids are going to get strung together from the code and produce the protein. The two COVID vaccines that hit the market first, the Moderna and the Pfizer, have packaged messenger RNA for a protein that is expressed on the surface of the COVID coronavirus. And so it gets your ribosomes to make the protein, and there's a couple of other fancy things there because the protein needs to get expressed on the surface of those cells that are producing it. So first, you got to get the messenger RNA into the cells, which is very tricky because a lot of times stray nucleic acids are read by your immune system as an indication of viral invaders. And so we're pretty good at finding foreign nucleic acid strands and just cutting the hell out of them, just breaking them all up. So they had to package the messenger RNA in a little lipid capsule that not only would make it more or less undetectable in circulation, but also it attaches to the membrane, and that's how the mRNA gets into the cell. Now, my issue with that as an approach for a vaccine, and I've been digging and haven't been able to get an adequate answer to this, is that, okay, here's your protein. It goes onto the surface of your cell as a foreign molecule, an antigen. And then your immune system goes, that doesn't belong. Let's make antibodies to it. That's the whole purpose of the vaccine. You make antibodies to the antigen that's on the virus by expressing them on your cells. When you get your shot, the cells that are accessed are in the muscle and in the drainage system, but there's enough vaccine that it also gets into your general circulation. And my question has been, so what cells are picking this stuff up and making these antigens? And if there's a set answer, nobody's telling, which to me is somewhat telling. And I'm a little worried that long term, but now I haven't taken either of those viruses. I'm waiting for the adenovirus, which does this a very similar process, but on lining cells in your respiratory system. And those cells are going to wear off. And that's my question. So you have cells in your body expressing these antigens. Are they doing it temporarily? Is it there relatively permanently? What cells are they? Because at some point, when your immune system is built up to fight the virus, it's going to recognize those antigens as foreign and go after them no matter where they are. If they're on cells that you kind of would like to have hang around, like cells in your nervous system or cells in your muscle, then do you really want your immune system attacking those? And I'm a little worried about having these weird random autoimmune responses as a long-term side effect of these mRNA viruses. And it may be completely unfounded. It's just weird that in the original research for these types of vaccines, there were particular target cells, dendrocytes, in your immune system that were going to express the molecules. Doesn't seem to be true with these two vaccines. And the persistence of the molecule on the surface, nobody's really talked about that. Nobody's really said, oh, these are the only cells that are going to be invaded and express the antigens, and that kind of worries me. And I'm going to get that out there. I don't want to get it out there too widely because I don't really want to get the people that are anti-vaxxers, because I may be totally wrong about this. It's just that based on how this all works, it makes some sense to me that there could be some ramifications that somebody had to have thought through. But typically, if somebody has thought through possible downsides, they'll address that. You'll be able to find them going, oh no, that's fine, we're doing it this way, so that it won't have those kinds of downsides. And I have seen people in the business also having discussions kind of amongst themselves about the same issues, like what cells, how long, that sort of thing. So it isn't just me, but it's a technical enough issue that it hasn't really risen to the surface anywhere that I've seen, except in technical discussions. Anyway. So, for whatever reason, you need a protein. You have genes, codes for that. Sorry, not on the screen. You have codes for that on your chromosomes. 2, sometimes you have more than 2, sometimes there are duplication events and you have several. But you've got codes for that particular protein. And so how do you turn that particular code on? There are activators. So a messenger molecule will go into the nucleus looking for a little stretch on the DNA that is a marker for that particular gene. It isn't the gene itself, it's near the gene. And it actually starts the unspooling and unwinding that you need in order to get at the gene. So those are called activators. And once you've got the system kind of activated, it's spelled with an E or an O, yeah. There's a whole bunch of ancillary molecules that are involved in this. The whole list is not always involved, but they're fairly important. You've got promoters that will mark just exactly where the gene starts at on the DNA. So the activator is, okay, come over here, we'll get the code, the DNA opened up, and the promoters are, and you're gonna start what you need to start right here. Okay, here's a partial list of what else you get. Let's see. There are initiation factors to try to get this rolling faster. There are stretches that kind of separate out the gene from the rest of the DNA, boundary elements, sometimes called insulators. And sometimes there are stretches on the chromosome, but it's not necessarily that close by. But it does stuff to the chromosome that makes it easier to work with, and those are enhancers. And I am oversimplifying the hell out of this. But just to give you an idea, this is a lot more complicated than it sounds like. And this is a really simplified version of what's going on. So here's your DNA. This side is the side that has the actual code on it. That's the other side that you use when you're making a copy. It gets opened up. This has been moving along it, and it opens up, separates, makes a copy, lets it close up behind it. This is RNA polymerase. And this is little mRNA, messenger RNA. Here's an A on this side, there's a U. Remember the pairing? G and C and C and G work. T and A work, but if you have an A on the original DNA, what you're gonna wind up with on the RNA is a U. Right? That's just because of they're very similar molecules. There's pictures of them in the book. You'll see that they're very, very similar molecules. This is called transcription. Where this starts, okay. We talked about codons being 3 nucleic acids in the sequence that codes for the amino acids. And I kind of sort of said that this was on the DNA, but technically that's not where it is. It's kind of like mitosis, being used in one way, but actually applying specifically to something else. The codons are actually messenger RNA, are these 3 nucleotide sequences in the messenger RNA molecules. And there are a whole bunch of them, and they code for different amino acids. But there's one at the beginning, so the messenger RNA gets made. there's a sequence called the start codon that is also a code for methionine. Because once you started, you can use that code for methionine. And also, so far as I can find out, that's the first amino acid at the beginning of the strand, is that the strands always start with methionine. It may get clipped off at some point, but it tends to be there right at the beginning. And I could be wrong about that, but so far as I can find out, it's not just a start codon, and that's all it does at the beginning. It's a start codon and also puts methionine at the start. And it goes until it hits a stop codon, which is basically, this is the end of the code. The messenger RNA may continue to get made for a while after that. But that's where it's going to stop making protein when it feeds through the ribosome. Here's the problem I wind up with. As I say, there are details to this. It's much more, we're simplifying it, and I'm never sure exactly at this level for majors that are going to wind up having to know the bits and pieces of this. How much of the bits and pieces to fit in, how simple to keep it versus how complicated it can be. But just as another picture of the same thing. Again, here's an A on the DNA side and a U on the RNA side, but where there's a C, there's a G. Where there's a T, there's an A. Where there's G, there's a C. And here's the RNA coming off. If you know your English, you might give somebody a recording that they're gonna convert to, they're gonna type it out. That's transcribing. You transcribe an interview, for instance. You're not changing the language. You're just changing it from one form to another. You got a nucleic acid to start with, you're making another nucleic acid. They decided that was going to be transcription. It's another picture of the same thing. Just kind of showing the whole thing with the promoter marked. RNA clips on, kind of has a little area where it's opening and making the copy. That moves downstream until it hits, they're calling it terminator DNA. There'd be a stop codon right at the end there, and that it's releasing, which it may or may not do, depending on the code. There are many times where you want to get a hold of whoever names something and say to them, why the hell did you do that? Sometimes the whole sequence that you're making is not going to be used to make the protein. You're actually going to adjust the sequence. You're going to snip pieces out. Sometimes you're going to snip pieces out and stick them together in an order different than they came off of the code. But a lot of times they're just going to be snipped out and then spliced together. There's what they call RNA splicing. And there's sequences that are never going to be part of the code that you use. And here's where I got a problem. Everything that's going to be part of the code is called an exon. Everything that's snipped out and not going to be used is called an intron. Now, to me, I'd have named them the other way around. Cutting it out, exon, leaving them in, intron. That isn't the way. It's one of those things where, to me, I remember the logical way to do it and then remember, oh no, it's actually named opposite to that. But this allows some flexibility where you can use the same genetic code to make different proteins because the splicing is going to be different. And sometimes the splicing is screwed up, and that's the root of some genetic diseases where things are supposed to get spliced and then reattached, and they don't get spliced and reattached. So your code winds up being different from the code that it was supposed to have. And of course, there's a whole bunch of functional RNAs that are involved in this inside the nucleus as well. Okay, now the messenger RNA, see, they do have this marked as a tail. The messenger RNA is headed outside of the nucleus. And so that's the end of transcription, and we'll pick this up in the next video.